This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 187, recorded on Friday, June 8th, 2012. Hi, everybody. I'm Vincent Racaniello, and you are listening to TWiV, the podcast all about viruses. Joining me today from North Central Florida, Rich Condit.
Hello there, Vincent. How are you?
Hey, fellas.
Hey, fellas. Well, there's only one of you. What am I going to say? Hey, fella? I could say howdy. That's another one.
Hey, fellas. Okay.
Howdy.
Howdy.
Okay.
How you doing? How you doing, Rich?
Good. 79 degrees, overcast. We had a lot of rain. There's stuff coming through, but we're starting Florida summer here. I got up this morning and it was at 6 a.m. It was 74 degrees and 96% humidity.
Wow.
You live here for a while and you learn to love that stuff.
That's impressive.
Yeah, it's pretty cool.
Well, I happen to be in Washington, D.C., where it is 28 degrees and really beautiful, sunny, just a few clouds in the sky. Very nice day. Everyone is remarking. Do you want to know why I'm in Washington, D.C., or
sure.
You
Vincent,
don't care?
Vincent, I care. Why are you in Washington, D.C.?
I'm doing TWiV Live from the White House.
Right.
Yeah, you're right. I'm kidding. You know, I am not too far from the White House. I'm here at ASM, American Society for Microbiology headquarters.
I've been there.
Which is a beautiful old brownstone type building. I'm in Chris Gondian's office. I was here for a meeting and I didn't have time to get back. So
does this meeting have something to do with like podcasting or that kind of stuff?
It is the planning meeting for ICAC, that meeting you and I went to last year.
Yes.
And
right.
Which will happen again this September, this time in San Francisco, though. So I am very close to the White House, and I was looking at my Google Maps the other day. Did you know that the White House has a basketball court?
I'm not surprised. I wonder if it's been there. I wonder if it's... so you can see it from Google Maps?
Yeah, if you go to Google Maps, and you could zoom in on the White House. So here I can see it because it's only a few blocks from me. And it says here the White House, and then if you zoom in a little further... the White House South Lawn, and it says the White House basketball court. It's really pretty funny.
Let's see. I'm seeing the Kennedy Garden and the Rose Garden.
Yeah, so if you go south of that, to the left, so there's the big lawn behind the White House, you know, with the circular drive, or maybe it's in front of the White House. It's on
south.
It's on.
I see a pool.
West Executive Ave, Northwest. You have to zoom in quite far. White House basketball court. It's a little white square.
It's not labeled on my map, but I see a swimming pool.
Yeah, I don't see the pool on mine. You know, the South Lawn. Do you see the South Lawn?
Oh, here we go. All right, here we go. White House basketball court. I see it.
There you go.
Is Obama playing?
I don't know.
He likes to shoot hoops.
Does he?
Yeah, this is quite a yard.
It's a big yard.
There's a nice swimming pool. Up north of there is a nice swimming pool.
I don't think he probably enjoys it all that much. Kind of a busy guy.
Yeah, busy guy. Jeez.
Anyway, so this is TWiV from Florida and Washington, D.C.
Cool.
We have a couple of follow-ups from last week. John Doe writes, I presume that is an alias.
I'm guessing.
The Monty Python Dead Parrot sketch came from the TV program. And he gives us a link to a video on YouTube and also the Wikipedia page for the there's a page for the Dead Parrot sketch. Originally known
there would be on Wikipedia.
A popular sketch or one act from Monty Python's Flying Circus, one of the most famous in the history of British television comedy. There you go.
This one, I watched it, and I confess I did not laugh.
Did you laugh when Bob Lamb said it?
No.
On TWiV? No? Okay.
Because I didn't get it, because I'd never seen it before.
Hey, maybe it's British humor, I don't know.
Well, there is something, no offense to my British friends listening, but there is something different about British humor.
Yeah.
Which is often hilarious. But in this particular case, like, have you seen A Fish Called Wanda?
Oh, I love it. It's wonderful.
That is one of the funniest movies I've ever seen.
Absolutely.
And John Cleese is one of my heroes. But the dead parrot thing did not get me somehow.
Did you see, right. Since we're talking about movies, did you see My Cousin Vinny?
Oh, absolutely. That's one of my all-time favorites. Yeah. Marissa Tomei.
Yes.
You just got to love it. You know, her mother tried to cure her of her Brooklyn accent when she was growing up because she figured that would be a problem. And it turned out to be an Academy Award winner.
I like it because it's My Cousin Vinny.
Yeah, that too.
Joe Pesci, right?
Joe Pesci. Great. Terrific.
Famous line of Marissa. Yeah, you meld.
Yeah. I never seen a grit.
I never seen a grit. Wonderful. All right, we just digress too much. Speaking of digression, check out the comments for TWiV 186, where there's a very nice discussion of stump grinding.
Yeah.
If you want to learn more about it, we mentioned that last time. Okay, let's move on to virology.
Do we have to?
No, we could just jab. I bet we could. I bet we could for an hour.
Yeah, I'm ready.
The first paper, I found this paper this week. I think it's pretty cool. It is amazing. It's a title. It's a paper in hepatology. So you figure if paper in hepatology, a virology podcast, it must have to do with some hepatitis virus, right? The title is Tracing Hepatitis B Virus to the 16th Century in a Korean Mummy.
This is great.
This is just so cool. All right, let's just review hepatitis B virus a bit. We did do an episode, right? We did an episode on Hep B. What was it? I had my number here. 80. Oh my gosh, that's a long time ago.
It's a long time ago. I can't even remember.
Michael Bouchard, I think you were on the episode, right?
Yes, I was.
It's called How Much X Could a Woodchuck Chuck? Anyway, Hep B is a double-stranded DNA virus. It's unusual because the genome is gapped. It's not completely double-stranded. It's an envelope virus, and it infects people. There are kinds that infect a variety of animals, but in people it's a serious liver infection. 350 million people globally are carriers. So you can pick up this virus and have an acute illness, liver inflammation, vomiting, jaundice, but it may also turn into a chronic infection where the virus is with you forever. And that can lead to cirrhosis and liver cancer. And many, many millions of people are at risk for developing liver cancer globally.
And if you're chronically infected, you may or may not experience recurring symptoms at various periods in your life. You can be completely asymptomatic as a chronic carrier.
Right.
Probably relevant that it's transmitted by blood transmitted. Okay, so very commonly in this day and age, intravenous drug use.
Right.
But it's also transmitted sexually.
Yeah.
And transmitted perinatally from mother to infant during birth.
Precisely.
And that's a serious problem because the probability that it will establish a chronic infection is very significantly higher if it is acquired by an infant at birth. And that's one of the reasons for administering the vaccine immediately when a kid is born, is to lower the incidence of that sort of chronic infection.
I think someone asked us about that once, why they should
they did.
Get Hep B.
That's right.
Good reasons.
We had a discussion of that, yeah. And actually, I think it's probably, although it's two other things come to mind, that one more relevant than the other. First, it's extremely hepatotropic, and that comes up in this paper. So although I think we've had some discussion of the possibility that it may infect some other tissues as well, it really homes to the liver, and I don't think anybody knows quite why. The receptor hasn't been identified. So whether it's a receptor issue or whether it's a replication specificity issue, I don't think is totally, at least I don't understand it. I don't think it is understood. And the other thing is that a particular bit of weirdness is that even though it's a DNA virus, it replicates through an RNA intermediate using reverse transcriptase.
Yes.
So it's a retrovirus with a weird sense of timing.
Yeah.
Even though it's unlike retroviruses in that the genome is never deliberately integrated into the chromosome.
Right. So the RNA is packaged into the virion in the cell, but it gets reverse transcribed to DNA before the virus particles leave cells.
Right. And then when the DNA re-enters the nucleus, it's transcribed by host polymerase into RNA without any integration step, and then you're launched.
No integration. Really interesting life cycle. Check out that TWiV 80. There's more there. So there are 10 genotypes.
Small genome. We're talking what? 3.5 kb? It's in this paper.
Yeah. We'll get to it. I don't recall offhand.
But small genome, just a few genes. We're talking a polymerase and a capsid protein and some accessory proteins.
10 genotypes, which are geographically restricted to some extent. Right? Korea, which is the topic of this paper, is endemic for Hep B and has almost exclusively genotype C. Over 90% of the 95% of the isolates are genotype C, and in fact, specifically C2, there are sub-genotypes.
As I read through this classification stuff, what comes to mind is that there are a lot of splitters in this field that are genotyping and subgenotyping viruses, but it's probably all justified. It's fine.
Yep. What is the origin of Hep B? There are two theories. That one is there's a primate origin. It originated in primates and went into people. Or there is a common ancestor for human and primate strains. So can't distinguish between those two possibilities. Mm-hmm. In this paper, they had this, well, I guess there are quite a few mummies in Korea that are studied, and in a separate paper, they showed how you can study these without invasiveness. You can basically do a CAT scan on these mummies and do a lot of sophisticated reconstruction and actually make amazingly accurate reproductions of what's inside the wrapping. So you don't have to unwrap them or disturb them in any way. That paper is amazing, isn't it?
Right, and that's important because there are ethical issues with invasive procedures on a mummy,
right?
Depending on who you are, where you're located, that kind of stuff. Apparently, these mummies also were not. These are mummified remains. They were not. They're naturally mummified, as I understand it. They're wrapped, but they weren't. It's not like there was a deliberate attempt to preserve them. Some circumstances of the burial ground is what wound up with them being preserved, so that there were actually soft tissues. Reasonably soft tissues remaining inside.
Right. You can see on the cat reconstructions, there's brain tissue, there's internal organs, and among those is what they think is the liver.
Right.
Brain tissue actually still has the surface structure of a brain. It's very interesting. Mm-hmm. So this mummy was dated to the 16th century by carbon-14 dating, which is the Joseon dynasty. I'm not up on my Korean history.
Neither am I. Did you ask your son about this? He seems to know all about this kind of stuff.
I didn't. No, I've not seen him since I began reading this.
Okay.
So what they did was to put a laparoscope into the liver, or what they thought was the liver, and pull out some samples, which they then used to amplify hepatitis B viral DNA. Now, I suppose one question they don't address is why they did this. Why? I mean, they had brain there. They could have looked in brain for other viruses. I suppose Hep B being so prevalent in Korea, that was one reason.
Yeah, you're right. They did not rationalize that, and I wouldn't be at all surprised if they aren't looking at other tissues for other stuff.
They say
if you're gonna isolate a virus
right
from a mummy, Hep B would be a good candidate. You know where to go to look for it. If there's a chronic infection, it would be pretty prevalent. It's DNA, so it has a better likelihood of sticking around. It establishes a chronic infection, so the DNA would stick around. So I suppose if you wanted to get a virus, I'm making this up as I'm going along here, but if you wanted to get virus sequences out of a mummy, Hep B would probably be one of the likely candidates for something that you could get.
Good thinking there, Professor.
Well, I'm paid to make stuff up.
I think those are good reasons. The RNA viruses would probably be hard to find, although I would look anyway. You could look in brain for herpes viruses. So, they're chronic and their DNA as well, I suppose. Right. This was a child mummy, by the way, 4.5 to 6.5 years old, and they did not know the cause of death. So this is somewhat of a long shot. But they, in fact, were able to sequence the entire viral genome by amplifying with primers, right, from these materials.
Yeah, they go through some discussion of that. They would prefer to do it by direct DNA sequencing, just deep sequencing or something like that. But apparently, because anytime you go to amplify something, you're at risk of either getting mistakes in the product or actually amplifying the wrong thing, as we well know from our extensive discussions on XMRV. So it's a tricky business if you're going to do PCR to amplify something. However, in particular when it comes to ancient DNA, I guess it's damaged enough so that the direct sequencing didn't work well. Among other things, cytosines have a tendency to go off to uracil, and that complicates things. They oxidatively deaminate to uracil. That's going to come up later, actually, in a letter. So they decided to do it by PCR, but they have an extensive discussion in here. Actually, they refer to a paper about how to do PCR of ancient DNA right, and there's all sorts of precautions that you need to take to make sure that you don't amplify some contaminant. And they even go so far as to say that at least in one of the places that everybody was seronegative for the virus.
Right.
They did it at three different geographically separated sites, one of which had never had hepatitis virus anywhere near the laboratory. So they were very careful to make sure that they had independent verification of whatever they got.
Right. Korea, the UK, and Israel, 3 quite distant labs, and they said they used clean facilities without plasmid. They didn't use positive plasmid controls, so they wouldn't.
You do the DNA workups in a different lab than you actually do the PCR. The PCR is done in total isolation,
right,
etc.
So they end up amplifying the entire genome, and it turns out to be a genotype C2, which is the prevalent genotype in Korea, but it's unique. It's different from any contemporary sequence. Any virus that's been sequenced that's circulating now in Korea doesn't have this particular sequence. I think it's 18 amino acids different from current.
Right, here it is, 3,215 base pairs.
That's the length of the genome.
That's how big it is, 3.2 kb. And so what's that? That's about a potential, although there's overlap in this genome and some codons or some regions are used more than once, that amounts to what? 1,000 amino acids, so 18 out of 1,000 is not a lot.
Remember, this is about 600-year-old mummy, right? So I'm surprised that it's so conserved. Of course, there are probably lots of non-coding changes.
DNA is good stuff, dude. I mean,
yeah, that's true. It is DNA. Much lower mutation frequency.
So that's 1,000 amino acids. So 18 out of 1,000. I'm doing my math here.
Now, while you're doing that,
at 1.8%.
Okay. So people may say, well, you said there was an RNA intermediate, but that is produced by host RNA polymerase copying a DNA template, so it's not terribly error prone.
That's a good point, actually. But the RT,
yeah, that's error prone. Yeah,
the RT is error prone, so that's a good point.
But that only happens once, right? It's not like it's producing tons of genomes.
Yeah, that happens once, but the RNA polymerase doesn't proofread. So, I mean, so what happens is that the input DNA gets copied into a whole bunch of RNA copies, and then each of those gets reverse transcribed once to make a genome. But neither the RNA polymerase nor the RT are proofreading enzymes. I mean, it's basically, with a different timing, it's basically the same process as HIV. And HIV goes through all sorts of variations. So, yes, I will now agree with you and say I'm surprised that it's that stable.
I'm looking up mutation frequency in hep genome. There are quite a number of studies, and I'm waiting for one to load here just to get an idea of what this might be. Anyway, so it's a C2, and they make a very careful point to say we don't know what's going on in North Korea for obvious reasons.
Right.
So this may be different there if you looked at hep B viruses there, so we can't really say. Okay, let me see here.
They say they've got something in the paper because they talk about they say HBV mutation rates per site per year. So that's not talking about per rep, or less than 2 times 10 to the minus 4th to 4.5 times 10 to the minus 5, but not exceeding 10 to the minus 6. That doesn't really tell me a whole lot, and I can't.
I got a paper here where they say the mutation rate is tenfold higher than other DNA viruses.
Okay, so that makes sense.
Because of the RNA intermediate. That makes sense. Back to the paper. The sequence, when you compare now the sequence to all the other sequences we have, it suggests that this particular virus, they call it ancient hep B virus, evolved at least 3,000 years ago. Remember, the mummy is about 600 years, and maybe longer. And they actually suggest that this originated in China or Japan because they were able to do genotyping on the mummy and predict that the individual came from China or Japan. So maybe that virus came from there as well. Or is this...
so they know that from like mitochondrial DNA or something like that.
Yeah, so they STR partial profile matches human haplogroup D of an Asian origin, probably from Japan, supporting the thesis that the mummy-derived ancient viral genome originated from Chinese or Japanese communities that migrated to Korea.
Interesting.
Yeah.
So one of the things that puzzled me about this is, and I guess I didn't understand some of the data about divergence.
Mm-hmm.
Is it seems to me that they use published figures on the rate of divergence to try and figure out how old this thing is. But it seems to me that I would want to do this the other way around. And here I have a virus that is related to the modern C2, and I know it's 400 years old.
Mm-hmm.
It seems to me that I could use that information to verify or refine the amount of drift in these viruses. Maybe I don't understand correctly how this can be done, but I would do it the other way around.
I think they did use it they sort of calibrated their clock based on this,
Okay.
related to all the other known sequences, right? So
Okay.
you know how much difference there is between these and contemporary sequences. You know how many years are in between, and you kind of can figure out, the clock, and you can tell if that confirms the clock or not, and then you extend back, and that's where they get.
So they're thinking this 3,000. So they think that this particular strain arose on the order of 3,000 years ago.
Yeah, and
saying?
So they say this sequence evolved at least 3,000 years ago, probably earlier. And one of their estimates is maybe 100,000 years ago.
Right.
So in other words, Hep B existed in Asia at least 3,000 years ago, maybe as long as 100,000 years ago. Probably more, because you remember we did a paper where they found Hep B DNA integrated into the finch genome, and that dates it millions of years ago. So probably as long as people have been around, maybe even pre-humans had Hep B. But you can only say so much based on these data, right?
Right. Wouldn't surprise me if it had been around for a very long time.
Yes. So basically, this is just amazing. This is the second ancient viral genome, but this is now the oldest ancient viral genome. Professor Condit, what would have been the oldest ancient viral genome?
Oh, no.
Before this one?
I was going to ask you. I wasn't quick enough.
Well, you can ask me. It's fine. You want me to?
What was the oldest ancient genome that had been sequenced before this?
Well, that would be the 1918 Spanish influenza virus genome.
Right. Okay.
Which you remember was derived from dead people and from slides from lungs. Similar idea, except that was an RNA virus. Mm-hmm. So now here is about a 600-year-old mummy. So this is the most ancient genome. I suppose you could even go further back with the Egyptian mummies, right?
I suppose. Could look at some of those for the same thing. I wonder what else you can find in these guys. I'm sure we'll hear more.
Yeah, this whole field is, just getting started. In the microbial world, the bacterial world, of course, people get teeth from mass burial grounds that were made in the plague era, right? And they can find plague bacteria in them. The pulp of the teeth is a good place to look because it has blood flow, and then it's preserved, right? Once you die, the tooth is preserved and it's not contaminated. So there are a number of groups who do that and pull DNA out of there. It's quite interesting.
One of the, I was going to mention, one of the controls they do in this generally a control, is to sample another tissue and show that, or state that they can't find any Hep B DNA in that tissue.
And what was that tissue, Professor?
Was that brain?
It was lung. It was lung.
It was lung.
Yeah, it's more proof that this is, not a contaminant. But it's so different from any contemporary sequence that it's probably good enough.
Right.
So this is cool.
So cool.
Don't you think it's neat? Mummy DNA. You ever see the movie The Mummy?
No.
No.
I went when I was in, I think, about 6th grade. Went for a sleepover at Skip Farina's house, and he wanted to stay up and watch The Mummy, but his father wouldn't let him, and Skip threw a fit. I was just as glad because I figured I was going to be too scared. Boris Karloff was the classic monster guy. I'm going to have to get this movie, and there's a modern version as well. I haven't seen that.
I've seen the modern version. It's not as good. Not as good.
So you've seen both?
Yeah, I saw the other one when I was a kid. It's really good, and the modern version is not very good.
I think I actually put it on my Netflix queue. I'll have to do that.
Okay, the next paper is a paper about what's happening today, and this is a paper that came out in Cell. The title is Personal Omics Profiling Reveals Dynamic Molecular and Medical Phenotypes. Now, I probably wouldn't have picked up this paper, except that I heard our friend Mark Pelletier interview the author, the PI on the paper, Mike Snyder, on Futures in Biotech, episode 93, I think that was. And I thought, this is such a cool paper. And as you will see, there is virology involved in it. There's also an article in the New York Times about this study, which is pretty cool. So the idea here is that, we've talked before about how many individuals feel that sequencing the genome is going to be really helpful in medicine, but the genome sequence isn't enough because, often diseases are multi-gene and there are environmental contributors, so you need to have other data before you can really interpret a genotype. So what they did in this study is they took an individual and they, over a period of over a year, forgot the number of days exactly.
14 months, something like that. And actually, depending on how you count it, 14 months to 2 years. But the actual data that they're reporting was over a 14-month period.
And they sequenced the genome of this individual at incredibly deep levels, in other words, high coverage, so they could really get accurate sequence. And they actually revealed some new sequences that nobody had before.
And they used two different technologies to approach the sequencing. And he makes the point, I haven't listened to the whole Futures in Biotech podcast, but I've listened to some of it. He makes the point that using a couple of different technologies is really helpful because they have different types of error that show up, so that's self-correcting.
Right.
When you compare the two.
Yeah, it's a great discussion they have on Futures in Biotech. So in addition to the genome, they did his transcriptome, which basically you take messenger RNAs and you sequence them.
Right.
It's also called RNA-seq these days, I think, right?
Right.
And this is
you only have to do the genome once, but the cool thing about the RNA-seq was they did it at several time points over the 2-year period, so you could see changes.
Right, because that is mRNA, which changes in abundance depending on time and tissue as well. Although here they only looked at peripheral blood mononuclear cell RNA, right?
Right, because it's easy to get at.
Yeah, take blood. But as you said, you would have liked him to be hacked up and...
Yeah. Go for it. I mean, if you're going to do the experiment, lung, liver, brain, we ought to send the paper back.
The subject of this study was none other than Mike Snyder. And it's interesting in the paper. They say that a 55-year-old healthy male, but in Futures in Biotech, they revealed that it was, in fact, Mike Snyder himself, which is pretty cool. If this were my lab, I'd do it on myself as well.
Absolutely. And he talks about, in the Futures in Biotech episode, he talks about why.
Yeah.
In terms of, basically, my guess is that the IRB thing IRB is easier to process if it's you. But most importantly, there's no problem in making sure that the subject is going to follow up with the study. He's not going to bail out.
Right.
If it was somebody else, it might be hard to recruit and might not necessarily follow through. And then he said there's lots of people in his lab who would have liked to participate, but that is unethical. So they were excluded from participating.
So we got genome, we got transcriptome. They also did proteome, which means you isolate all the proteins, right? And you determine their levels.
Right, and that's done by mass spectroscopy.
Right. Now, that's a smaller database than the others, but in fact, they found that it was sometimes unique and told them things that they didn't get from the other information. Right,
and as he points out in the paper, in some ways, that's, well, I guess it depends on your perspective, but in some ways, since proteins do most of the real work, in some ways that's a more informative profile than the transcriptome. Right. On the other hand, they do microRNAs as well. There's a lot of non-coding RNAs around that are doing a lot of work. So
right.
It's 6 to 1, half a dozen the other.
And the final thing they do is the metabolome.
Now, I didn't understand the difference between the proteome and the metabolome. The proteome, the only difference I could see is that it looked like the proteome was a protein extract of the peripheral blood mononuclear cells, whereas the metabolome was a protein extract of just the cell-free serum.
Well, I think...
And the processing was the same. It was mass spec in both cases.
Yeah.
Is that right, or am I missing something?
Let me look here. I don't remember. I thought the metabolome was a variety of enzymatic assays. But let's see.
I think you have to go to the supplementary material to get at that level.
Yeah. I can do that. I think I have it here. Let's open it up. Because when you go to a physician, they take blood and they run, a variety of profiles. As I said on Futures in Biotech, about 20 or 30 different tests, which are pretty limited typically. But he said, here we can do thousands and thousands and thousands of different things. I mean, the idea is that one day this will be done on everyone. I'm not sure you could do it at intervals like this is done, but Serum Metabolomics, here, 100 microliter serum sample. Metabolites were extracted. So they're looking at actually metabolites by liquid chromatography. Various things in the blood, not just proteins, but the other components that might vary.
You can certainly look at lipid and
blood glucose, all that sort of stuff.
Vitamins, all kinds of stuff.
Exactly.
Okay.
So that's what that is.
All right.
Okay, so what brought this to my attention when I was listening to it is that he had 2 viral infections in the course of this study. The first one was right at the beginning, so he calls it day 0. And he had a rhinovirus infection. So basically, the way I view this, I actually wrote him an email and asked him about this. He must have had some sniffles, and they said, let's take some of my snot and see what virus it is. And so they got rhino, they typed rhino out of his nasal secretions, a swab, I think. And they said that at day 0, but in fact, the infection probably began before that. Right, because when you have symptoms, of course, the virus replication is actually already decreasing in a rhino infection. You understand what I'm saying, Rich?
Yes, I understand what you're saying. But I never understood. I was baffled by this notion of making day 0 the rhino infection, because then they have a time point at day minus 123. So I guess that's probably samples that were taken when he had his genome sequenced or something like that they could use as that's the only other earlier time point. Is that right?
Yeah, I think at minus
123.
So minus 123, he had a physical to make sure he was healthy.
Mm-hmm.
And so that's why they set that. And then I don't know why he set it.
They must have just decided when he started feeling sick, okay, we got to take some time points here. And ultimately set that as time 0.
Let me tell you what he wrote. All right, so I asked him, he said, for the first viral infection, I was sick on day 0, so undoubtedly I was infected before that. It was when we first took blood. Yes, it was a nasal swab. For the RSV infection, we actually called the first day I started seeing symptoms day -1, minor congestion. Day 0 was defined to sync up with the HRV infections. Probably not the best nomenclature.
Mm-hmm.
So that's Mike Snyder's response. So he started at the beginning. They decided, all right, we're going to start this. And he had a cold. The day he comes in the lab to start this study, he's got a cold. So it's just coincidence. So he had a rhino infection for a while. And then it's actually day 0 to 21 they have it here. And then at day 289... he had a respiratory syncytial virus infection. So again, he had sniffles. They took a nose swab, and they had RSV.
To me, the notion that he's got an RSV infection is interesting all by itself, because RSV is a virus that causes severe. Usually, you catch it in childhood.
Yeah.
And can cause a very severe respiratory disease, though not necessarily, but it can. But the immunity is poor and not long-lived. So this is it'd be interesting to look at his, I wonder if he could look at his, probably he's got enough serum samples to see whether he had RSV antibodies before this thing took off, because probably that's not a primary infection. Probably that's a reinfection.
I would bet, yeah, because that's what we teach, is that you get it as a kid and you get less and less severe symptoms as you get older. But maybe, I don't know, maybe his antibody titers were low, whatever, yeah. Before I forget, I want to focus on the virus part, but before I forget, so based on all his genome sequence, they run these programs where you can do riskogram, and you can calculate your risk for various diseases, which I guess you're going to be able to do, Rich, because you have your genome sequenced, right? You're having it sequenced.
Right, it's being sequenced. Yeah, I would hope somebody sends me a riskogram someday. And in Futures in Biotech, he has this long discussion about, whether you want to know this information.
Yeah, it's a really good discussion.
Which is interesting.
Yeah. So he's at risk for a variety of diseases, which he sort of discounts. But two of them are quite interesting, and this page has such a huge figure on it that it scrolls really slowly. Where is the riskogram? Here we go. One of them is type 2 diabetes. Mm-hmm. And he's like, I'm not diabetic. I'm not even the profile for a diabetic. And he went to his physician, and on the podcast physician says, no way, I'm not testing you. You're not the classical diabetes case,
No family history, none of the risk factors.
I think it's a mutation in, where's the mutation? I forgot where it is. Insulin receptor, I think. Yeah.
Yeah, I don't know.
Something like that. After the RSV infection, he became diabetic.
Actually, I think there are a number of risk factors here. This riskogram.
Yeah. The highest one is dyslipidemia, right?
Mm-hmm.
Glaucoma, basal cell carcinoma, coronary artery disease.
Oh, I'm looking at part C here. It was used to calculate the risk of type 2 diabetes. And he talks, there's, 20 or so different alleles.
Right.
Or SNPs that factor into that, calculating that risk factor. So it's not just one thing, it's a bunch of stuff.
Basically, when he got the RSV infection, his serum glucose shot up. We have a chart here on figure 2D, and he became symptomatic, and that's when. So he thinks that maybe the mutation combined with what the virus did in terms of inducing genes somehow pushed him into high blood sugar. So he modified his lifestyle and brought his sugar down. But it's interesting because he would never have picked this up until it was really serious, probably.
So I'm wondering, first of all, whether he actually experienced symptoms of type 2 diabetes. I mean, you can see it in the glucose profile, but I don't know that he actually felt it.
I don't think he did. Yeah, I think they picked it up by these lab tests.
And so he did lifestyle changes and all of that kind of stuff, and it brought it back down. But I wonder if it would have come down anyway, because it correlates. It goes up. It correlates with the RSV infection. They're not sure that the virus infection did that.
Right.
Could be some other cofactor, because it lags the virus infection a little bit. But you have to wonder whether this kind of stuff doesn't go on all the time in people with his genotype. That you get some sort of infection or something like that, and you get a spike in your glucose. Maybe you feel feel a little crummy for an extended period of time, and then it just resolves itself spontaneously.
Yeah,
I don't know, but I wonder.
I think he did admit that in the podcast. Yeah, that they can't really, they wouldn't know, but he decided to make these lifestyle changes. He changed his diet, he started exercising, and so forth.
It's one of the problems. You can't do the controls.
I mean, look, if you got this data and your serum glucose were up, you'd probably do the same thing, right?
You bet. It's exactly what I'd do.
All right, so then here, let's just talk about the virus infections a little. They did because of what they were doing, they have a complete profile of the transcriptome of what is this, serum, I think? Yeah, serum cytokine profiles. That's figure 2F, where you got different colors for going up and down. You can see at day 0, the rhino infection, many of the cytokines are red. That means they're shot up.
Right.
And they go back down to normal. And then day 289, the RSV infection, again, the cytokines shoot up. So that's exactly as we would predict, right?
Right.
And then, of course, they can also look at genes that go up and down. So RSV infection, 2,000 genes. This is gene expression. The transcriptome went up. 2,200 went down.
There's another thing about this cytokine profile.
Yeah. And
that is, if you look on the bottom there at day, I think it's 301, he has a little arrow pointing with a question mark. Mm-hmm. And as I was looking at this, I noticed that it's as if there's a second spike.
Yes, it's like 18 days after the first.
In the cytokines.
He mentions that in the text.
Right. As a matter of fact, that's 18 days after the infection. That's when the glucose goes up.
Yeah, maybe
day 329. No. The glucose goes up later. That's day 329. This is day 301. But he doesn't. I don't recall that he comments on that question mark specifically there. But I noticed it, and it seemed to me like. Maybe this happens. Maybe it's not exactly the way you would predict, that there's bumps in the road for this immune response. On the other hand, it could be that something else happened that's unidentified that spiked his cytokines again.
Yeah, he notes this spike on day 18 post-RSV. And he says those genes are in pathways including the spliceosome, glucose regulation of insulin secretion, and various pathways related to a stress response. Mm-hmm. So something happened there. Who knows if it's related to RSV or not. What I found
shortly thereafter, his glucose shot up.
Shot up, yeah. What I find interesting is you can sort of get an idea of the number of genes that go up when you get a virus infection. A few thousand, basically. I should say transcripts, right? Both
in the peripheral blood mononuclear cells, okay, which are primarily cells that are going to be involved in. They're going to be involved in the immune response. This doesn't say. RSV is a respiratory infection, right? So that's infecting your lung tissue. So it doesn't say what's happening in the cells that are actually infected with the virus. This is relevant to the immune response to that infection.
This is a good point. You can see that there is a global response, even though this is a respiratory infection, right? You've got changes in your serum, which is everywhere, of course. It's very interesting. Right. So what kind of genes go up? Immune system genes, stress-related genes, phagocytosis genes, insulin, B-cell signaling, immune processes of various sorts. It's just as you would expect. Major histocompatibility genes go up. It's really interesting. Let's see, he calls
then they have all of this analysis that looks at pathways. This is becoming a real bioinformatics is, you can make a living doing this stuff now.
Yeah, sure.
There's interactomes, right? All of these proteins interact with each other, or by one criterion or another are on the same pathway, and there are bioinformatic programs that allow you to say, okay, if I've got these 10 things going up, are they related to each other, and does that indicate that I've turned on a certain pathway? And these analyses can address that point.
Exactly. So they say overall the transcriptome analysis captures the dynamic response of the body to infection. And of course they add the proteome, which they find some new things that they didn't detect, and they also find confirmation of the transcriptome results, especially in with respect to virus infection. It's pretty interesting. Let's see, anything else we should point out here? Yeah, I think the coolest thing for me was that a couple, 1,000, 2,000 genes go up and down when you get a virus infection, at least in PBMCs. I think that was it. It puts a number on it, because you're always wondering how many genes change. So a lot of things happen when you get an infection.
Yeah, it makes sense to me because, among other things, there's going to be a lot of signaling going on. Just to get the cytokine response and get all of these immune cells collaborating with each other to deal with this infection, there's a tremendous number of signaling pathways that go on and a lot of communication from one cell to another. 2,000 genes doesn't surprise me at all.
Right. And among these genes, go ahead.
It's interesting to consider that, and this was a point that was made in a seminar that I attended some time ago and made really nicely, that not only do you have to turn these genes on, but you have to turn them off.
That's right.
Right? You can't just turn them on and leave them there. Turning them off is an active process as well. Otherwise, among other things, these immune cells proliferate in response to an infection. And once they proliferate and they've done their job, you got to get rid of them. Otherwise, you got a lymphoma every time you get sick. Right?
That's right.
So those are both active processes, and it's fascinating.
I should also say that there were some genes turned on by viral infection that were surprising to me, and I could see people going on to study these now and trying to figure out what they do in a virus infection. I don't remember offhand what they were, but I was thinking, well, it would be interesting to look at that in cell culture and see if you get the same effect.
Right.
So the idea here is that eventually in people we'll have these kinds of databases of us, and they'll be periodically updated. And you'll be able to monitor your health this way and pick up things.
Well, it makes sense to me that you might go in. You might go in for your annual physical.
Yeah.
And you've got your genome sequence on file, and they'll take blood. And when they take blood, they won't just assay a few things. They'll do deep sequencing and look at your transcriptome, and they'll do all of this other stuff and get some assessment of your situation in time at that point. And it could be that you'll go in and you're not feeling too well, and they'll do the same thing and get some idea of what's wrong with you.
Right.
And we're talking maybe 50 or 100 years from now when it's cheap and worthwhile. But
the price does have to come down, but I think it's probably going to be 10 years because things go down really quickly in price. Yeah. But you're right. Right now, this study, he said, is just prohibitively expensive. And, of course, the genome that he did is at huge coverage. Right. I don't remember the X coverage he did, but it's really much higher than you would do on normal people. Anyway.
Well, he calls it a proof of principle.
Yeah, that's right.
Which is exactly what it is. It says, okay, we can do this, and we can get information out of it. Actually, the type 2 diabetes thing is important because without something like that, you might kind of go ho-hum. But he makes the point that the genome sequence predicted that he was predisposed to type 2 diabetes, and then lo and behold, he has an episode that has a profile of type 2 diabetes down the road. So he's saying that, the genome sequence really was predictive.
Yeah. It's just incredibly coincidental that he had that episode. Mm-hmm. I wonder how many times that happens in people with this mutation.
Maybe it's not that coincidental. It could. Yeah. Maybe it happens pretty frequently, and you just never know it.
That's right. We've never looked at this level. You're absolutely right. Maybe if you have this particular mutation in this gene that made him at risk for diabetes, then if you get a virus infection of a certain kind, your serum glucose spikes. And then it goes back to normal. Who knows? Really interesting.
His genome sequencing was they did complete genomics at 150-fold coverage and Illumina at 120-fold coverage.
Yeah, that's a lot.
And exome sequencing, using exome sequencing, what is that? Is that the RNA-seq? I don't really understand what exome sequencing is. Using 3 different technologies at 80 to 100-fold coverage.
Exons. It's for sequencing exons. Yeah, so it's mRNA.
Okay, so
right?
that's RNA-seq. Okay.
Yeah. At
any rate, that's really deep. And as you say, they came up with new sequence information that's not in the reference human genome.
Right. So if you can't read the paper, listen to Futures in Biotech 93, and we'll also put a link to the New York Times article about this. It's really, really cool stuff. All right. Shall we do some email?
Sure.
Can you take that first one there?
Sure. Don writes, "Thank you all for sharing your knowledge in such a comprehensible manner. Thank you also for your stand against bureaucratic censorship in the H5N1 research and your win. I have 2 questions. Is H5N1 a highly specific test for a human genotype/phenotype? And if those who died of the disease were sequenced, would that give a good estimate of the true risk for the rest of us? Question 2 concerns the genetic code. A, G, C, T, and U are the reasons of physics or chemistry that make this the only way to archive information, or is it just Mr. Darwin's best shot to date? And why is uracil? Is it a chip off the old Rosetta Stone? Again, thanks so much for your hard work and excellent podcast." I don't understand the first question completely. Is H5N1 a highly specific test for human genetics?
So in other words, those people who die of H5N1, do they have some mutation that predisposes them to serious disease?
Okay.
So in other words, are we selecting for those people?
Okay.
You know,
All right.
And I think we've brought this up at Northwestern briefly with Bob Lamb because we had a related question. I think that those individuals probably have a predisposing mutation for sure, and
Right. Okay. So I remember that discussion. Yes. Good question.
It's not been looked at, but I bet if you did genome sequencing of those individuals, you might get some clues. But we don't know because they are typically in places where, we don't do that or it's not done yet. So, but I bet that they are particularly susceptible for one reason or the other.
And we do know, and I forget which TWiV this was, we do know that there are mutations that can predispose people to severe flu infections.
That's right.
We did one of those
Exactly.
just a couple episodes ago.
That's right.
All right, good question. This other one I really like.
Yeah.
And I don't know if I've got this right, but check me on this, Vincent, because so why A, G, C, T, and U? Well, I don't know why those 4 bases, okay? They're probably, yes, Mr. Darwin's best shot to date, and we're pretty locked into that. But the interesting thing is why thymine in DNA and uracil in RNA. And one of the problems here is that, see if I can get this straight, there is a tendency for cytosine to spontaneously oxidatively deaminate. And when it does so, it goes to uracil. And so DNA-based organisms have derived a mechanism for dealing with that, which is to evolve a system of enzymes that recognize uracil in DNA and repair it back to cytosine. Okay? So
you can't have U in DNA, basically.
U in DNA you can't have. But that raises the question, why do you have U in RNA? And I would say it's because RNA is short-lived, so you don't have this problem of getting the deamination. And second, that was it was originally an RNA-based world,
right?
So you could get rid of it. And then you evolved the whole thing with Cs later on. I'm not sure that makes complete sense because the changes from U to T.
From U to T
but you can't have U in DNA.
To go from U to T is a methylation, right?
Yes.
So if we first had an RNA world with a U, so say U just evolved, maybe those enzymes that, probably the enzymes that repair it to C were not existing at the time. So it could exist in an RNA world. And then we evolved into a DNA world.
Okay. Yeah, right. We evolve into a DNA world. If you put U in the genome and you've got C in the genome and the C deaminates to U, you got a problem.
Yeah, right. So you got to get rid of the U. Yeah.
So you got to get rid of the U. So you can't have U around. So you create the enzymes that take care of that, which means that you can't use U anywhere. You better use something else, and so you use T.
Yeah.
Okay.
Yeah.
So yes, that makes sense.
It's cool. It's pretty cool, isn't it?
Oh, so complicated.
The use of AGCTU, I think they happen to be pretty good because they can form, they do the complementary thing. They can form this extended chain linked by the riboses, and all that. I think that just probably there are other ways you could do it, but this is what's evolved, yeah. But it does. Right. There are specific requirements. Remember, there's base pairing and the formation of very long molecules. So maybe not every compound can do that. That's a good question.
The whole thing is amazing.
Next one's from Gerald, who writes, "Hey TWiVites, I'm a 32-year-old low-waged data entry clerk without a college education, and I've been unsure of what to do with my life over the past decade. It's not until I listened to science podcasts like TWIS and TWiV that I began to reevaluate what I want in life. Your podcast jump-started me into going back to college to get an associate in arts for teaching secondary biology for high school or middle school. Your engaging and insightful conversations gave me something worthwhile to strive for, where otherwise I was a little lost. I thank all of you for the interesting conversations and for keeping it on a level that the general public can follow for the most part. I'm particularly happy when Alan joins the group. I've always enjoyed his stark, careful logic when approaching a perspective or problem brought up in the program. Anyways, keep up the good work for us fans, and I'll be listening down in Miami."
Terrific. We touched somebody else's life in a big way, apparently.
It's great.
Yep.
Thank you.
Well, you're quite welcome.
You're
Glad
welcome.
to help.
Yep. Rich.
William writes, "Good day again, TWiV doctors. The ethics statement in the Sim et al. Paper is geared more towards, 1, the use of animals to maintain the colony outside of experimental infections, and 2, using the mice in the host-seeking assay. The mosquitoes used in this study are a long-colonized lab strain and are typically maintained generation to generation by feeding on an animal, often a mouse, although chickens are also used. Hope this provides a little clarification." So that was TWiV 179, the paper where they looked to, what was this? This was dengue infections in mice.
Makes mosquitoes thirstier for blood.
Right. Right.
They did the transcriptome of the mosquito, and they found that dengue induces genes involved in perhaps odorant detection and feeding behavior, that sort of thing.
That's right.
So I think we were talking about the animal protocol and what they were needed for, so that's what that's about.
Right. And so the animals are used not only for the experiment, but to maintain the mosquitoes outside of the experiment, right?
That's right. All right, thanks, William. William is a graduate student in New Mexico. Kevin writes, Dr. Racaniello and compatriots. Something that has come up again and again in discussion over things like chronic fatigue syndrome and influenza is sampling and the different medical measures considered. I just saw a paper published by some computer security researchers at Microsoft who discuss some fundamental sampling problems. Their focus is computer crime statistics, but the problems described apply widely to other fields. and make an interesting read. 1. A representative sample of the population doesn't give a representative picture of the surveyed quality. 2. Outliers can cause catastrophic errors. And 3. For rare phenomena, we are measuring a signal weaker than the noise in which it is embedded.
Mm-hmm.
And it has a great title, Sex, Lies, and Cybercrime Surveys. He provides a link to a PDF of that. For what it's worth, I'd love it if you had an episode with some computer security folks to chat about statistics, responsible vulnerability disclosure, and possibly also viruses.
We've talked about getting a computer type on here before to talk about viruses. These would be other good things to talk about as well.
Yeah. I think it's an interesting point. I mean, these issues about sampling, I think we touched on a little bit when we talked with Michael, the epidemiologist, Michael Walsh, right?
Mm-hmm.
But they are good points, and I think there is an overlap in viruses of computers and animals and other living things.
Yeah, it'd be interesting to get a computer jock's perspective on that. All right. Jeffrey writes, doctors, how many more pieces of biological machinery are byproducts of viral infections? To quote the narrator in old Tootsie Pop commercials, the world may never know. But I would like to suggest that the venom genes in reptiles and platypuses are just such a mechanism. I admit that I know little about the evolution of the venom systems, but a gene sequence search might just show some close matches with either viral or bacterial sequences. It would certainly be fun to find out one way or another. So this refers to the notion that maybe cells have copped some viral genes to use for themselves, and the best example that we know of is the syncytians grabbed from retroviruses that are important in implantation, placenta formation.
Right.
So he's suggesting maybe venom genes. I think he's thinking of like virulence factors or something like that brings that to mind.
I think this came in after we did the wasp parasitoid episode.
Uh-huh.
And so, the wasp delivers these virions that have wasp genes in them into the caterpillar. Right. So he says maybe venom is the same thing. But it's a good idea. You could do a search targeted at finding that out.
Mm-hmm. Venom is really complicated stuff. It's got all kinds of stuff in it. I didn't know that platypuses had venom.
Yeah, they have that little spike underneath one of their rear legs, and they could put venom in with that, yeah. I used to be fascinated with that when I was a kid. Next one's from Robin. Dear TWiV team, podcast 179 was amazing. The wasp using integrated viral capsidization genes for a vehicle to deliver its own immunomodulatory genes to target caterpillar cells is nature more imaginative than the best of science fiction. It compels a further rethinking of basic concepts as to what organisms really are. Cladograms of the tree of life overlook the fusion of mitochondrial lineage with an anaerobic lineage to produce eukaryotes. Viruses are paraorganisms that alter properties of cells. Their integration into cell lineages is a reminder that nucleic acids are the players, and we are epi phenomena after several layers of emergence overlying them. That includes all of biology and all of what we call civilization. Well, this is very heavy to me.
That's very heavy. But the notion is that what I read in this is the notion is that nucleic acid replication is what's driving the whole thing. And we're just pawns.
Yeah, that's right.
In that whole thing. And I don't disagree with this.
The selfish gene, right?
Yeah, absolutely. And we think we're all important in that kind of stuff, but we're just stooges for DNA trying to replicate itself.
Right. Very cool. I like that, Robin. Thank you. Thank you for that.
Yep. Tom writes, Dear Dr. Racaniello and colleagues, sorry I couldn't think of a more clever TWiV appellation that hasn't already been used. As always, I'm very grateful for the show. I have a small suggestion that might help spread TWiV to more people. Many sites and online articles use buttons to automatically create links that can be shared on social networking sites like Facebook or social news sites like Reddit. Reddit especially has a large community of science-appreciative lay folk just waiting to get infected by TWiV. These buttons could appear at the header for each week's episode, along with the retweet button you already use. I would also suggest these for Alan Dove's blogs. His recent entry on bee colony collapse disorder was great, and I think it's the kind of thing that should be spread online to as many readers as possible. Also, I'm writing to have a throw at my thoughts on the Fahrenheit Celsius issue. I completely agree that we would all be better off using a more metric system for weight, distance, etc. But I'm very partial to the Fahrenheit scale for describing the weather. While 0 to 100 in Celsius perfectly describes the range of temperatures we use in liquid water-based chemistry and biology, 0 to 100 in Fahrenheit neatly caps the extremes of temperature we experience in most weather. 100-point scales make sense to me. It just depends on what you're describing. Anyway, looking forward to your upcoming 200th episode benchmark. TWiV is a great show, and it's really influencing how science can be communicated. Best regards, Tom.
I think he's got a point about the Fahrenheit. That's what you have said, right, Rich?
Well, I'm just used to Fahrenheit. Okay, and actually, that as I understand it, that was not developed. The extremes of that scale, 0 and 100, were not developed based on weather. It was 100 was supposed to be body temperature, but when he measured it in his armpit, I think, and it turned out that when we understood what real body temperature was, it was not 100. Or that seems a little backwards because armpit would be lower in temperature. At any rate, it was inaccurate, and I believe that 0 was the lowest temperature that he could get by adding salt to ice water. Okay, it was the lowest temperature he could get for liquid water in a solution. I think that's right. So it's not necessarily based on the weather. It was just sort of a couple of weird extremes. Right. But I'm just used to it. And it is, I suppose, coincidental that in terms of weather, 100 is about my limit.
Yeah, it's true. It is true.
But it makes more sense, actually, in terms of weather to me, that 0 is when freezing happens.
Right.
Whereas on Fahrenheit, 0 is what? Minus
32.
Something, minus 32.
Yeah.
And how about these buttons?
Yeah, we can do buttons. I don't have a Reddit button, but I do have other buttons at the bottom of each post.
Yeah, I found that, I guess that's at the bottom of individual episodes.
Yeah, you have to go into the individual episode, and maybe a lot of people don't do that.
That's what I've found, is that if you just look at the TWiV site...
Yeah, you don't see the buttons, right?
You don't see the buttons for each episode. You have to click on the individual episode, and then you see them.
So I will fix that, and also I've been told by a number of people that Reddit is what we should be targeting, so we'll put a Reddit button as well.
Okay.
All right, the last one is from Nina, who writes, this question is not entirely to do with viruses, but I really enjoy your podcasts and couldn't think of anyone else who would have scientific theories. It's very flattering, but there are plenty of other people out there. I've been reading a lot lately about two different topics. One, how diet and stress impact epigenetic regulation, and how, for example, a bad diet may impact gene regulation and lead to disease states. Two, the paleo diet and other plans that insist grains are bad for your health, mostly stating that since the eating of grain evolved only, say, 10,000 years ago, humans are not well equipped to digest, and this can lead to poor disease states. I can't find any scientific evidence towards these last claims. It doesn't seem really logical. This would mean that any agricultural process is not healthy for us. It does seem healthy to cut down sugar intake. especially all of these high-carb beverages out there today, and to limit grains. But to cut these things out altogether, what is your scientific opinion on how what you eat impacts health? Can a bad diet lead to a higher susceptibility to a cold or maybe even cancer? What makes up a good diet? There seems to be so many opinions and non-scientific claims out there, it's hard to discern fact from fiction. Thanks for any thoughts on this. Well, I think you are right on, Nina. I think this paleo idea, if there's no proof for it, it's just a theory.
Right.
I mean, the point is there's no evidence that this is the case. This is a theory, right? And I personally am fine with grains. How about you, Rich?
Yeah, I'm all over grains. I think it's great. She said a key sentence here that I really like is she says, "I can't find any scientific evidence towards these last claims."
There you go.
So
there you go.
She's questioning stuff, and that's great.
Yeah, don't accept
that's
it. Yeah.
Absolutely. Yeah, that's absolutely terrific.
And it's true. It is true that a good diet is absolutely essential for defense against infections.
And diet can influence other things as well. I'm thinking of some cancer incidence studies where there will be a higher incidence of cancer of a particular type, say in, I think I'm thinking Japan, or a lower incidence in Japan. And so there's some question as to whether or not there's a genetic component there. But if you look at generations of people of Japanese descent that are raised, for example, in the United States, you find that with respect to certain cancers, they assume an incidence of cancer that reflects the U.S. population rather than the Japanese population. And the suspicion is that there are environmental factors, notably diet.
Right.
Okay, so I don't know that you can point to a particular diet, or maybe you can. I'm not well enough informed, but I think that there is good epidemiologic evidence that diet can have a profound influence on health.
I think, societies with high-fat meat diets like ours have a high incidence of colon cancer. And that's not the case in Japan. They have other types of cancer based on what they're eating. So you asked about what you should eat. So the NIH actually has recommended a particular kind of diet. It's called the DASH diet, Dietary Approaches to Stop Hypertension. This is meant to lower high blood pressure without medication. And it's high in grains and grain products, fruits, vegetables, low-fat, non-dairy foods, etc. And it's also known, has foods from this so-called Mediterranean diet. So I'll put a link to that, and you can check that out. It's not,
and that has scientific evidence to back it up.
Exactly. I mean, this is not a fad diet. This is science-based dieting, and I think it's quite good. But the point here is that a lot of grain and grain products are recommended. So there you go. So much for the paleo diet.
I was not aware of this DASH diet. That's really good to know about. And it's not, I mean, the word diet in this is, we're not talking about this is what you do to lose weight.
Right.
We're talking about this is the way you ought to eat to maintain good health.
Right.
And I think beyond that, how much you eat of this particular balance of nutrients is going to influence your steady state weight.
Exactly. All right, we got through all the email. How about that?
How about that? Is there not other email lingering out there, or are we actually through it?
No. We have plenty more. I got about 100 others waiting.
Wow.
But don't let that stop you. We'll get to them and just keep sending them in. Okay, what do you have for our pick of the week, Rich?
I have for a pick of the week. We've been talking about checklists at various times, and I have referred to this article on a couple of different occasions, but I thought I would actually make it formal. This is a 2007 New Yorker article by Atul Gawande, who himself is a physician, talking about the use of checklists in medical practice. And he talks about some studies done by, going to have to look at this, Peter Pronovost, who is also a physician, who's been advocating the use of checklists in various procedures in medicine, and has done a number of studies to show that they are enormously helpful. In particular, one of his primary studies was using a checklist just for putting in a central line in somebody. You're supposed to gown the person and use a disinfectant to wash the site and wash your hands beforehand and blah, blah, blah. And he found that if he instituted a checklist so that this was done right all the time and empowered the nurses to call out the doctors if they didn't do it right, that he could reduce the incidence of central line infections by some phenomenal amount, like 75% or something. And it saves many, many lives and all sorts of money. And part of this also is a little bit of history on how the checklist evolved, which we've mentioned before. It was evolved by aviators to make sure that they don't crash their airplanes because they're so complicated. And he's got a little piece on that. Very well written, very interesting article.
It's terrific. It's just.
And I've been thinking about this. You know, I use checklists basically in a casual fashion in my own work. When I do an experiment, I write a protocol.
Right.
And in particular, the experiments that I do now, these protocols are like 20 steps long or something like that. I check them off as I go along because I can't remember from 1 minute to the next whether I've done whatever it is.
Right.
So
I do the same thing. We all do checklists. Sometimes we have them in our heads, or sometimes we write them down, but it's important to do them.
Just the other day, I didn't follow my checklist, and I did something wrong, and I had to abort a whole experiment, and it cost me a week.
All right, you learned. There you
go. I learned for a little while.
Yeah, I'll screw up again. You'll do it again.
Yeah.
My pick of the week is the website of a painter called Michelle Banks, excuse me. Now, a couple of weeks ago, actually a long time ago on TWiV, we talked about a artist who makes virus models out of computer parts. And a few weeks ago, he emailed us and told us he was going to have a show in Arlington, Virginia. So I'm here in D.C. On Wednesday, I went to see the show. I saw his display, which is an adenovirus model made of CDs and hard drive parts. But he had also said, go see Michelle Banks. She has virus-themed art. So she was on the same floor. I went to see her display. It's very cool. She has a website, and that's what I'm linking to now. And she makes cool images, watercolors, viruses, bacteria, cells, cell parts. In fact, I was so enthralled, I bought a piece of art of hers, of viruses, which I'm going to hang in my office. And in fact, while we were recording this podcast, she dropped it off here at ASM for me to take home. So,
oh, that's cool.
Yeah, I bought it yesterday, and then she emailed me. She said, "Hey, if you're still here, I live in D.C. I'll bring it to you." So, I got it.
So, which one is it that you bought? Did you post this somewhere?
I didn't post it, but I'm going to post it this weekend. It's off her site now because when you buy something, it disappears because they're all one-of-a-kind pieces. But
that's what I was going to ask. If these are all original watercolors, this is not like a print or something like that.
No, each one is original.
No kidding.
So you buy it, it's the only one, and it comes off the website. It's very cool. So she seems
what
wonderful.
Inspires her to do this? These are all, they're cool.
She said she just loves small things in biology. You know, she loves microbiology and cells. She's not a scientist. She just loves and is fascinated by the natural world, especially at the microscopic level. You know, if you sent her a virus picture, she'd probably do a print or a coloring for you of it. Of course, you'd have to buy it from her, but she's always open to inspiration. So check her out. It's really cool.
This one of Petri dishes is really cool.
Yeah, it's very nice. A lot of cool stuff.
Oh, yeah.
So I'll post pictures of the exhibit this weekend and link to it because I took pictures of her stuff and also Forrest's, who does the virus sculptures. They're both really cool. So check them out. Support them if you can because these are artists. They need a little support for their work. We have a couple of listener picks of the week. One is from David, our friend at Vassar. Since the topic of personal genomics has come up a few times lately, yeah, even today, right? I thought this might be a good pick of the week. It is a NOVA program on ethical and other questions raised by personal genomics. How ready are people to know their own genetic code? What would you do if you find out you are susceptible to heart disease? Or what would you do if your genes suggest you are not? And as you saw today, this is exactly what this genomic study raised for Mike Snyder. So that's pretty timely.
I think some of these ethical issues and personal issues are very interesting.
Right.
And they're not going to go away.
And we have a second one from Josh. Dear TWiV doctors, here's my pick of the week. It's a 2009 editorial in Bioterror and Biosecurity entitled The Nuclearization of Biology is a Threat to Health and Security by Franz Ehrlich, Casadeval, Imperiali, and Keim. Since Dr. Imperiali was on the show discussing these issues a few weeks ago, I thought it would be relevant. So there you go, and thank you, Josh, for that.
And this is a really nice editorial comment. And I'm impressed by the fact that all these guys are NSABB members, and they include a variety of disciplines, including this guy. France is a former commander, U.S. Army Medical Research Institute for Infectious Diseases, and Ehrlich is a lawyer, and of course Casadeval and Imperiali and Keim are scientists, and they say, be careful here. If we apply the same sort of regulation to biological research that we have developed for nukes, that we are likely to do more harm than good. And that, coming from them, that is comforting.
It is. It's very good. So thanks for that, Josh. That's really good. And that'll do it for TWiV 187. Don't forget, you can find us at twiv.tv or on iTunes. And we have an app for your iPhone or Android device at microbeworld.org. We love to get your questions and comments. Send them to twiv at twiv.tv. Rich Condit is at the University of Florida in Gainesville. Thank you, Rich.
You're quite welcome. Always a pleasure. I am now going to be absent for quite a stretch. I think it's at least 3, maybe 4 weeks.
Mm-hmm. We will miss you.
I'm taking off next week to see my daughter in Austin. Mm-hmm. Austin, Texas. That's a suburb of Buda.
Great.
And then I'm taking off abroad for a meeting after that. So I will miss you guys.
We'll have fun and have safe travels.
I will.
And when you're back, we'll look forward to it. We'll miss you.
All right.
I want to thank Chris Kondayan for letting me use his office here at the headquarters of the American Society for Microbiology in Washington, D.C. I'm Vincent Racaniello, and you can find me at virology.ws. You have been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.